Hello and welcome to Living with PSC, a podcast about living with the rare disease of primary sclerosing cholangitis. My name is Niall McKay, and today we are talking to Dr. Christopher Bowlus, MD, Professor and Division Chief of Gastroenterology and Hepatology at the University of California at Davis. Today we're talking about diet and some of the emerging treatments for PSC. In your own words, give us a brief description of the disease and what it is.
It's surprisingly difficult to actually explain what primary sclerosing cholangitis is, at least diagnostically. I think, typically we think of this as a disease uh, involving the medium to larger sized bile ducts and causing a fibrosis or sclerosis of those bile ducts, which then become obstructed and cause further injury or damage to the liver. And any damage over time to the liver eventually leads to end-stage liver disease, or what we call cirrhosis, and potentially to the need for liver transplantation. There are lots of conditions that can cause the same appearance in the liver, this so-called secondary sclerosing cholangitis. But primary sclerosing cholangitis refers specifically to a disease that's seen frequently in people that have underlying inflammatory bowel disease, more frequently found in men. And there are some unique genetic and immunologic features that we see in PSC. But it's not uncommon to have difficulty determining in sort of the atypical PSC patient whether it's truly PSC versus one of these other secondary causes.
Typically, is it cholangitis that sort of takes patients down, if you like, or fibrosis of the actual liver itself?
Yeah, so cholangitis means inflammation of the bile ducts, but oftentimes when we talk about cholangitis in sort of a generic sense, we're talking about an infectious process. So cholangitis can occur in people without PSC from any obstruction of the bile ducts. Typically, that would be from let's say, a gallstone that gets stuck in the bile ducts. Um, but the inflammation of PSC and the stricturing or scarring can cause obstructions that lead to infection and cholangitis, but those are usually not life-threatening. Treated with antibiotics and dilation of the strictures can generally relieve that. But what's most commonly the progression of the disease that leads to some problem, some life-threatening sometimes complications, is just the scarring that occurs and the failure of the liver, the complications with cirrhosis that then require liver transplantation. So that is, probably 80% of the outcomes, what we would consider clinical outcomes of PSC. That is something that either needs, treatment for liver transplantation or could even, lead to death.
And the scarring that occurs in the liver, that is more or less caused by either toxic bile, or it's caused by bile, either too little or too much bile. Is that correct? Or
That's the proposition. There's probably many different factors that lead to the scarring, and the problem we have in PSC is that in order to make the diagnosis, a fair amount of damage has already been done. There's already scarring or sclerosis of the bile ducts, so we're not even sure what the early events are that lead to the initial scarring. But clearly, abnormalities in the bile, which includes the so-called toxic bile acids, can cause further inflammation and injury to the bile ducts and the liver in general.
and anecdotally, is there anything patients can do to reduce the amount of toxic bile that gets into their system?
Well, that's a big question, and that's where treatments like ursodiol, have been studied and tested to try to um, uh, alter the composition of the bile acid. So ursodiol is in fact a bile acid that we produce naturally, and it has certain properties that make it less uh, injurious to the bile ducts. And so by taking that supplement, we sort of dilute out the toxic bile acids and the total amount of bile acids that our liver produces. Now, so far, the actual clinical data has been somewhat mixed and not really that strongly supportive of ursodiol for the treatment of PSC, though there may be a subgroup of patients that do benefit from it. Some of the newer therapies that are being tested now actually reduce the amount of bile acids produced by the liver, and that's another approach that may be effective. And then some of the areas that we're sort of investigating now would be actually dietary modifications that will alter the bile acids produced. So our body responds to our diet in order to produce bile acids that help us digest foods, and certain foods induce different types of bile acids. And then bile acids also affect the microbiome in our intestines and vice versa. Bacteria in our intestines act on bile acids and cause them to be changed. So there's this very complex interaction between the bile produced by the liver, the bacteria in the intestines, and the diet we ingest. So sorting that out and figuring out where we can intervene, I think, is an area of a lot of interest.
So, for the sake of argument, let's say that I'm sitting here with a nice fatty pork chop sitting in front of me, that's already making my mouth water even thinking about it, although I don't eat many of them these days. Um, what is the process? So I ingest the pork chop, and what then happens in terms of bile production?
So whenever we eat a meal, particularly a fatty meal, that actually causes release of bile and the bile acids that are in the bile into the intestines, and they help break apart and digest primarily fats and allow for their absorption. We've also found, though, that bile acids have signaling properties. So certain bile acids actually are bound by proteins on many different cell types, and they tell the cells to do certain things. And so the bile acids that are actually produced will cause differences in how much bile acids are produced and which bile acids are produced. Um, and so that pork chop you ate, if it's fatty, will cause a release of bile acids, and then depending on those bile acids, that composition may be changed the next time you eat. And in addition, those bile acids are going down into your intestine, further down into the gut, and they are being altered by the bacteria there and then reabsorbed and going back to the liver and recirculated into bile again as what we call secondary bile acids.
So we're not sure, I presume, whether it's the primary or secondary bile acids that are causing problems, or and this conversation that's taking place between the microbiome in the stomach and I guess there are hormones that are telling the bile ducts or the production of bile, is it?
Some of it is sort of a hormonal effect that is FGF19 in the mouse being released by the intestines, where bile acids are absorbed and going back to the liver and causing some impact on the amount of bile acids produced. But you're correct in terms of which specific bile acids, whether primary or secondary, are causing the most injury is something that's really not well understood at this time. We're just now getting to the point where we can find some associations between certain bacteria in the colon and bile acids in the stool, which may give us some insights into which may be most injurious, but we're still just at the very early stages. And this is all association. We don't know what is cause and effect at this time.
Would you like to talk about fecal microbial transplants as part of the treatments, or is this something that you've done any research in?
I personally have not done any research in FMT or fecal microbiome transplant or transfer. Uh, but the concept is actually quite old and was used several decades ago to treat patients with infectious colitis from a bacterium called Clostridium difficile. And that's where the excitement of this procedure has been really shown to be effective in many studies. And essentially, it's the concept of replacing a microbiome that is disrupted, usually by the use of antibiotics, killing off the good bacteria and allowing Clostridium difficile, which is naturally in our colons, to proliferate. And then by providing stool from a healthy, presumably healthy individual, reconstitutes the microbiome of the person with this C. difficile infection. And that's been very effective in terms of controlling this disease in patients that don't respond to conventional therapies. The thought was that in other diseases where there are alterations or what are called dysbiosis of the intestinal microbiome, excuse me, and this could be anything from ulcerative colitis and Crohn's disease to things like obesity and cirrhosis. The data that FMT is effective has not been that strong. In some ulcerative colitis trials, they've kind of been plus minus in terms of the effect. Some of the take-home messages, I think, are that there may be certain people, certain donors that have microbiomes that are particularly effective, whereas others may be less so. And I think that's one of the keys here is we're really just throwing in an unknown soup from a donor into a poorly defined recipient. And so there's a lot to learn there in terms of what we should be actually using to reconstitute the intestinal microbiome.
It's part of the difficulty for PSC patients, isn't it? Is that the treatments cause some of the problems in itself. So typically, if you get an infection, you take an antibiotic, and the antibiotic also may be killing some good bacteria in your stomach, so it might, increase the reoccurrence of the infections.
Well, I think the idea that antibiotics alter the natural microbiome, narrow the diversity. I mean, diversity is sort of one of the key concepts I think we've come to understand in microbiome is there are lots of things we do from diet and antibiotics that narrow the diversity of organisms in our gut flora. And so antibiotics potentially could be a trigger for either colitis. I think there's less data in terms of PSC, but there's some pretty good evidence in terms of inflammatory bowel disease that antibiotic use is associated with an increased risk. So they probably play a role. What's interesting is the concept that antibiotics might be effective in PSC, and how whether that's by altering the microbiome or other properties of these antibiotics is yet to be fully determined. There's questions in terms of when we intervene and what effect we might have. So, for instance, if someone with PSC has very advanced liver disease, just changing the microbiome in their intestine is not likely to have a big impact on their liver. On the other hand, someone very early in disease, in which the microbiome becomes, quote unquote, more healthy, that may have an impact on the disease in a significant way.
And do you think, like, just in terms of for people who are, do things such as, live yogurts and kimchi and, the things that encourage more stomach bacteria or more healthy stomach bacteria. Do any of these things make any difference at all? Or
The clinical evidence is not there, especially for PSC, where we're really lacking, anything that's shown good clinical efficacy. There's unlikely to be any harm from it, and I think that if people feel better taking probiotics, yogurts with active yeast, I think that's all fine and may have some benefit to it. As I mentioned, diet also is an important factor, both in terms of bile acids and the gut bacteria. And so a diet that is well balanced with lots of vegetables, unprocessed foods, does create an environment for a more healthy, diverse microbiome. So that is probably a reasonable approach to take.
And particularly, I believe some studies to show that green vegetables provide some help.
Yeah, it's providing more complex nutrients allows for a more diverse microbiome. And what we've typically see within, industrialized countries where there's more processed foods is, a less diverse microbiome. And it all goes along somewhat with this whole concept of the hygiene hypothesis, which has been around for many, many decades. The idea that we don't see a lot of autoimmune conditions in less developed countries, but as countries become more developed and bring in more meats and more processed foods, we do see a rise in autoimmune diseases, and particularly in ulcerative colitis. And the thought was that this was due to the fact that people aren't exposed to these bacteria early on in life, and therefore don't have tolerance towards them. And instead of having an immune system that is more tolerant to lots of different proteins and other substances they may be exposed to, they become a little bit more hyper alert. And so they're more likely to react to environmental triggers that they're exposed to later in life.
So, typically as well, though, there's a lot of antibiotics in meat, fish, and poultry.
Yeah, poultry especially, and yeah, that's it's not just antibiotics that we take through prescriptions, but it's also antibiotics that are in the food supply. I think we're getting away from that, but it's important to recognize that changes made today may not have an impact for a generation. Some of this, these things may likely occur, early in life or even in utero before we're born. Our microbiome is established at the time of birth, and there's differences whether we're born via C-section or a vaginal birth. And so while in an individual the impact may be very small, these small little differences in a single individual can lead to a disease.
Let's talk about some of the treatments then.
There's, to my knowledge, no dietary intervention studies in PSC to date. There are some data from the Mayo Group looking at dietary intakes and risk of PSC. So doing some food questionnaires on people with PSC versus people without PSC and looking for specific intakes of foods. And so things like grilled meats and such have been associated with a greater risk or more frequently seen in people with PSC. Similar to if you look at smoking, people with PSC are less likely to have smoked. So there's some association there. What we're interested in looking at is something called a specific carbohydrate diet, which is an interesting diet that was developed back in, I believe, the 1950s. And it's almost the exact opposite of what we might intuitively think nowadays, but it's gained pretty widespread popularity among patients with colitis, and it's been studied a little bit in pediatric patients. And so there may be a role to look at that diet. Other diets, such as a vegetarian or vegan diet, to see what impacts they may have. I think right now we're still at the early stages, though, and need a little bit more understanding of what are the effects of diet on the microbiome, particularly in patients with PSC.
But presumably, the more bile you need to produce, the worse off you're going to be at some level.
Yeah, but not just volume, but types of bile. And as I mentioned, I think it's hard to think that it's going to be just a one-to-one relationship. We have a very complex mixture of bile acids being produced. We have a very complex mixture of nutrients we're ingesting, and we have a very complex community of organisms that are interacting with those other very complex mixtures. And so trying to sort out this interplay of those three different factors is incredibly complex. And so I think the answers are not going to be so simple either. But I think as we look to this more, understand it, perhaps at excuse me, a sort of more global level, we might be able to get some clues into what foods might both alter bile acids and promote the bacteria that produce the processes that we want to promote. There are specific chemicals that bacteria produce that are absorbed and found in our body, things like butyrate that might be very good for at least our intestinal mucosa and maybe our bile ducts. And so those are things we might want to promote.
What's butyrate?
Through diet.
What is that? Butyrate?
Butyrate, yeah.
Yeah, and what is butyrate?
So butyrate's a chemical that's produced as a natural process of bacteria, acts as an energy source, and has other properties for some of the epithelial cells in the intestines.
And we produce it ourselves, do we?
But it's produced largely by bacteria in our intestines.
Okay. Sort of moving on just for a sec to the other treatments, the other new treatments there are a number of treatments that are kind of hormonal out there at the moment. Do you want to talk a bit about, or what do you think are some of the more efficient treatments coming out, or effective treatments?
Right. So if we talk about more traditional medical therapies, drug therapies, there are a number that have completed phase 2 studies, and a few others that are either completed or near completion, and so we should have some data on them soon. And I would say there's a lot of promise here. The traditional way of approaching these diseases has been through modulating bile acids, and that's where the most success has been. So about a year ago, a trial in Europe looked at something called nor-urso, which is very closely related to urso, but has some properties that suggest it might be more protective to the bile duct cells than just regular urso. And that study showed significant improvements in liver tests in patients that took nor-urso compared to those that had placebo. And that study is now going on to what we call a phase 3 clinical trial in Europe. And so hopefully in the next several years, we'll have results of that study and see if that's effective in slowing the progression of the disease. The other ones are obeticholic acid, which is like a bile acid that binds to one of those receptors that we talked about earlier, and it reduces bile acid production by the liver. And that showed significant improvements in the alkaline phosphatase in patients that took that medication compared to placebo.
I'm sorry, what's it called again?
Obeticholic acid. It's currently approved for the use in primary biliary cholangitis, or PBC, where it was found to reduce alkaline phosphatase. The other study that has not officially been released in any publications, but they've had a press release, is the MGM 282 product, which is sort of a hormone that's produced by the intestines and acts somewhat in a similar manner to obeticholic acid, but reduces bile acid production, and it had similar positive effects.
This is this FGF19 that
exactly. Yeah. Yep.
Which is a hormone.
And there's so the main concept here is working through what is the receptor is called FXR, or farnesoid X receptor. And so there are other drugs in development that target that same pathway that are either in trial or may be in trials in the future. There is also a study that was completed that targets the inflammation, and we're waiting results on that study. And we all know about the fibrosis study of simtuzumab that was a very large, well-designed study, but unfortunately had negative results. But what was important about that study is it really showed us several, I think, important lessons from doing clinical trials in PSC. The first is that PSC patients are very eager to participate in clinical trials. This study was a 2-year trial, required injections of medications, required 3 liver biopsies, and had over 200 participants. And to think that individuals would agree to participate in such a rigorous study, I think really says a lot about the PSC community in general and their desire to find some effective treatments.
Well, the choice is not great for them. It's like,
no, I didn't want to use the word desperate, but I think we were all desperate to find something. And but it's truly a commitment to participate in a clinical trial when you may get placebo, and even if you get drug, you don't know if it's going to work or not. And the other thing we learned about PSC during this trial was how the disease progresses, what kind of events occur, because we have this group of patients that were studied very carefully in a clinical trial where there are regimented blood tests, liver biopsies, etc., where we can really have a better idea of what we can expect in a clinical trial moving forward. Because one of the problems in doing clinical trials in PSC is the disease fluctuates from week to week and month to month. So if you have somebody in a clinical trial and they develop a complication, it's hard to know if that was due to the medication or is that just the natural history of the disease. And so this trial will give us an idea of what to expect moving
And do we
forward.
have any idea of that
And
now? What the progressions are?
well, the progression on the liver biopsies was not too remarkable. But again, the problems with liver biopsies are that changes tend to be relatively slow over years. And again, and this is one of the challenges overall for PSC in terms of developing new therapies. We're we're now learning, looking at a wide um, uh, spectrum of patients with PSC, that the average time from diagnosis to liver transplantation is about 20 years. So about half the patients have a liver transplant before 20 years, but there's still half that have not had a liver transplant after 20 years. And if you do all the calculations to actually statistically prove that a drug is effective in PSC by reducing the need for liver transplantation, it would be a study of thousands of patients over many, many years. And so that's the big challenge for us is when do we think a drug is actually
But
effective?
can't we? Aren't we in a much better position to study this sort of thing now? Even by having, I saw there was a PBC app available that where people could study their diets, their exercise, their whole regime. But if you put that together, say with the actual medical data, which is now not only recorded but kind of downloadable, you could actually do a very effective mining of the data that would have been impossible even a decade ago.
Absolutely. So PBC is a little bit ahead compared to PSC. And so in PBC, we have generated these models where you can enter your age, certain laboratory values, and it will give you a prediction of your transplant free survival over 3, 5, 10, and even 15 years. And we can apply that to groups of patients as well and see, oh, does this drug alter our prediction? And that will give us some indication that that drug actually may be effective. Doesn't prove it, but it definitely helps support it quite a bit. The problem in PSC is, despite our attempts to generate a similar sort of model, we just haven't had the data collected to date to be able to do that on a large scale. We're in the process of doing that. As you mentioned, a lot of this data is available to patients and downloadable, but there are all sorts of legal and ethical reasons why we just can't freely collect it. It needs to be collected in a certain fashion, and it is a big effort, but that is ongoing. So there's several efforts ongoing within the International PSC Study Group. This is a group of investigators throughout the world, and we're in the process of collecting this data, both here in the
And
U.S. as
also, there's
well
some
as
good
in
work
many
being done
countries in
on that,
Europe.
not just with the PSC partners in the US, but other, patient advocacy groups, isn't there?
I know the PSC Partners is doing quite a bit on that. I've been involved in that. We have not been collecting um, uh, large amounts of data from patients because it's variable in what data they have available and just the efforts it takes to collect it. I believe Save John was also working on a similar project, but I'm unaware of where that's at. Uh, so those are the ones I know about. But clearly, there's a need um, to try to develop a similar type of model that we have in PBC and PSC. Again, though, it's a little bit more difficult because we also in PSC have many different sorts of outcomes that can occur that may be unrelated to the disease progression. The risk of certain cancers can occur whether you have cirrhosis or not. So it's not just the liver disease progression that we need to be able to model.
And also, to some degree, I think the symptoms are hard to model, aren't they? I mean, they're hard to collect because fatigue, itching. I mean, I guess they're they're not, or liver pain, dopiness, or cloudiness of thought. I mean, these are things that are hard to both. Everybody suffers from fatigue to some degree, particularly in this modern world that we're living in.
Well, I'm on my second cup of coffee. um, right. No. So symptoms is a whole nother area, and again in PSC it is very complicated because fatigue is probably the most common symptom, abdominal pain, and what I learned from PSC Partners Registry is how much insomnia or difficulty sleeping is an issue for people. I didn't realize it was so common.
And that's not
And on top of all
that's not
that,
necessarily related to itching either, is it? The insomnia is.
I don't think so. We haven't really investigated that, but it clearly was a concern for many people. And on top of all the PSC symptoms, you also have inflammatory bowel disease that may be quite active in people and causing similar other symptoms. Now, another nice tool that has recently been developed and came out of that simtuzumab study was the development of a PSC-specific patient-reported outcome. And so patient-reported outcomes are tools that we use to objectively quantify symptoms in a group of patients with a specific disease. And so fatigue, there are many different scales or tools we can use to quantify fatigue. Same with abdominal pain and other symptoms, but they may not accurately measure the fatigue that's associated with PSC or other symptoms. And so there's this new tool called PSC PRO, which is just the PSC specific patient reported outcome. And so that can now be used to quantify symptoms, and it can be used in clinical trials and other studies to see if there are certain things we do, interventions, that may improve these symptoms. Now, that's not data that's readily available in our medical records, not routinely collected, but it is a new tool we have to try to investigate the other side of PSC, which isn't just the lab tests and sort of the medical outcomes, but how patients are actually feeling.
If a family member of yours had this illness, what would be the recommendations that you would make?
Some of this depends on the stage of the disease. So I think the first thing is to get perspective on the individual's stage of disease and symptoms. So if I were someone that had early stage disease that is very little fibrosis, and I would get something like either a FibroScan or better yet an MR elastography, and my blood tests were pretty much normal, I would feel pretty good and make sure I kind of have that mindset that I've got years and years to deal with this, and I'm in this for the long haul. If I was more advanced and looking at a liver transplant very soon, I would be, making sure that I have other things lined up in terms of making sure that I'm being monitored for complications of my advanced liver disease, such as varices, etc. For all patients, it's very important to make sure that we keep not only an eye on the liver, but also all the other components. So if I've got inflammatory bowel disease, I want to make sure I'm getting that looked at every 1 to 2 years in terms of making sure I don't develop any precancerous lesions. I want to make sure that I'm getting my liver checked at least once a year, if not twice, with an ultrasound. And personally, I would get an MRI once a year. I'd make sure I check my bone density, make sure that's okay. And otherwise, just checking on my liver tests on a regular basis, depending on how advanced my disease is.
In terms of that, I mean, categorizing how advanced. So early stage, PSC would be maybe what your stage of liver disease would be a 1 or a 2. Is that correct?
Exactly. Yeah, that's generally the case. So we would consider stage 1 to 2 early disease, and stage 3 to 4 advanced disease.
In terms of numbers of say, of the alkaline phosphatase, you're talking about within the low hundreds for alcohol for early stage, and then what, mid to high hundreds?
Well, actually, so when we talk about stage, we generally talk about the amount of fibrosis, and that's different than the alkaline phosphatase. I have patients that have very advanced disease and very mild elevations of their alkaline phosphatase, and vice versa. There are patients that have very high alkaline phosphatase but very mild disease. So the alkaline phosphatase generally reflects the amount of obstruction or injury going on, but doesn't necessarily correlate with the amount of fibrosis or scarring, or even how someone's going to do. We do know that if you have a normal or near normal alkaline phosphatase, the rate of progression, the risk of progression is lower than if it's higher. But other things to look for would be if the platelet count's low, the albumin is low, the bilirubin's elevated. Those are all signs that the disease might be a little bit more advanced.
Well, I'm afraid that's all the time we have for this week. For more information about PSC and for a list of people and topics we'll be covering in the near future, please go to www.pscpartners.org. That's www.pscpartners.org. PSC Partners is a non-profit organisation dedicated to providing education and support to primary sclerosing cholangitis patients. There is information and resources on the website. There is a conference once a year. There is a private Facebook group that you can also join. Also, please join our patient registry. This is an anonymous database that provides research support to the medical community who are researching this disease. Thank you very much for listening.